Well, the trials and the studies with that are still ongoing, and they haven't fully reported. So I think the jury is still out with osteopath.
A few years ago there was talk of taking skin cells from people with Parkinson's, taking them back to a sort of earlier stage by treating them with various chemicals and then following the progress of the disease in a dish. Did anything come of that, do you know? Or was it just newspaper talk?
I think Bunya may be able to add to this. There are some groups which are using fibroblasts and things from Parkinson's patients in tissue culture for studies.
Yeah.
Yes, certainly people are doing that. Question.
How is this drug administered? Is it something that, if it came on stream as fed, we could think it's quite easy to administer, or is it something to be taken orally, or you inject it straight into the
body, not the brain, because it crosses the blood-brain barrier. But yeah, ideally you would have something like a tablet.
Yeah, this is already out. So it would just be like taking another tablet, and it's fairly got a long half-life, so that you wouldn't need to take. You probably need to take some tablets twice a day.
How did you choose this particular drug to use for this experiment?
The DCBG, because this one is a newly designed drug. As Claire said, that one of the other receptor types that has been looked at, which is the mGluR4, there are several drugs which are being looked at for targeting that one. The reason why we chose the DCBG is that the mGluR8s have a much more, how can you say, a much more defined distribution within the brain. Whereas I think the mGluR4 receptors are more widely distributed. So the potential for side effects are probably going to be less with the mGluR8. And also that this compound shows very good effects on this type of receptor. So it's a novel drug, and we think that it's probably going to have more selective effects compared to some of the other drugs.
Who actually made the drug?
Well, we buy this in from, was it Campbell's one? Yeah, so these are made by chemical companies, and you can buy this drug off the shelf for research purposes. But obviously it would take a lot more work for it to get licensed to go into man. They are taking one other of these class of drugs into clinical trials. I think there's a drug which works on the mGluR5 receptor. And I think that's gone in, is it for dyskinesias, or I think they're testing that for trying to reduce dyskinesias in Parkinson's patients. So that one is actually going into man.
So it goes through the blood-brain barrier?
Yes. Yeah. So the whole, the reason why we've known about these receptors for a long time, and it's been a bit slow to develop drugs to target them, is that most of the drugs bind at this site here. And unfortunately, the chemical makeup of the structures which bind there mean that they don't cross the blood-brain barrier very well. So for years, this area of research has had a bit of a stumbling block. But now they've synthesized new compounds, of one of which is this DCBG, which can cross. But also they're synthesizing other molecules which target this secondary part of the receptor, these allosteric modulators. And these are smaller molecules, and they can easily cross the blood-brain barrier. So now there is a new emphasis about looking at this area because of all of these new drugs are coming through now.
So, since you said that because of the binding, the compound that's in the drug is a very specific molecule, but I was wondering in general that, do you know of any nutritional ingredient or like any foodstuff that contains similar these potential substances, and
There's nothing at all.
and as a general like kind of bigger question is that, but because if you find certain substances that contain like relevant molecules, then you potentially have another scientific method to experiment with them by taking a larger human sample and like try them with particular nutritional regimes which are proven to be safe and see whether you see any progress. Do you know of any kind of clinical work that follows like a parallel experimentation with nutrition in this vein?
Yeah, I think a couple of the drug companies have screened all of their what they call nutraceutical compounds that they have in their libraries against these types of receptors, and there's nothing that actually binds to them.
Okay, so it's been tried. Okay, thank you. I mean, if you don't mind.
Yeah.
This is slightly tangential, and I accept if you reject to answer this. This is kind of relates to a kind of larger moral theme of research. I was wondering if you have a specific stance on the ethical issues around using animal models, whether you personally think that there's ample fair justification for using these, or whether you have any kind of moral attitude towards this that you think you want to share.
Well, I think everybody using animals has a moral duty to use less animals. And what we do here is that we firstly try and avoid animals at all cost. So what we use is human tissue quite extensively. This is why Parkinson's UK funded the tissue bank, because looking at the brains of people that's affected with Parkinson's disease is a very good starting material there. And also we do quite a lot of work on cell cultures. But you get to a point where you need to look at an intact physiological system. So you need to put it in an animal to see whether it gets into the brain and see whether it affects the similar pathways that they're affected in Parkinson's in humans. So eventually you have to get to that point where you have got solid justification for using the animals in research. And Claire and nobody likes using animals, but we have to because we can't put that compound straight into man, because that is also unethical as well and can be quite dangerous. You probably all heard several years ago about that study that was done at Northwood Park, where it's a very experimental compound that went into man very quickly, and quite a few people died in that study. So we're also covered by the Home Office with our regulations. So I have to have what's called a project license, and the government have to approve my project license. And on that panel, not only are there scientists, they're also lay people as well. And each person working in the group has to have their own personal license as well. And all the work here is overseen by the vets, so that we ensure that when we're using animals that there is the minimum amount of suffering that the animals undergo. But also, it's important to understand that whenever we do the surgery on them, we do the surgery exactly like you would do surgery on a human. So they have the same anesthetics, they have the same painkillers, we do the same aseptic techniques. So there is nothing different about how we would do surgery on a human. So the animal is fully covered by anesthesia. It has all the pain drugs to minimize any pain that they may be suffering at the time. And it's like, after a human has had surgery, we give pain-killing drugs, so for that initial period after the surgery. But it's like when I had my knee operation done, a few days after, yes, I wasn't fully recovered, but I didn't need to take the painkillers anymore, because you And also it's worth bearing in mind that in the brain there are no pain receptors. So this is why you can implant things like deep brain stimulators into patients while they're awake, and you can just have a local anaesthetic around where they're actually doing drilling work.
How much collaboration is there between your research team and other research teams on Parkinson's?
On Parkinson's, yeah, there's lots of collaborative projects that we do. Obviously from the tissue bank here, we're here to really act as a source to supply and pump prime research into Parkinson's using human tissue. So we collaborate with other groups doing different projects. I've got a group that I work with at our South Kensington campus. You know, they're making nanoparticles to try and use as drug delivery platforms. We work with groups at UCL looking at the sort of genetics of Parkinson's disease. We worked with groups internationally as well.
So they know exactly where you are with your research. Do you all know where you are?
Yes, so we go to conferences as well and share data as well, and it's also widely published because you need to publish the data to get it accepted. So most people know what you're actually doing, and also I think the Parkinson's UK publishes abstracts from the projects that it's funding, so most people can find out what we're actually doing. Well, there are.
Sorry, could you repeat the question? I couldn't hear it.
And you're talking about familial forms. Yeah, there's rarer gene mutations which cause some what's called familial forms of Parkinson's disease, where it definitely passes down the family line. But the majority of cases are what we call sporadic Parkinson's. So there's no definite family link there. But there are some families where it really does pass down, but they're quite rare, those cases.
In the future, would you be able to use this and want to see what the effect of the drug is in combination with the levodopa, or would that be too difficult to actually?
Yeah, that's one of the studies that we propose to do, is actually to give the compound with, well, first of all, see which dose of levodopa on its own alters the behaviour back to normal, and then to give the DCBG and to see how, if we lower the dose of L-dopa, do you actually still see the same response? So is it enhancing the effectiveness of L-dopa? So that's one of the things that we're going to look at.
Well, they do more stuff.
Yes, one.
Could I just ask, could you tell us a little bit more about the brain bank you have here? And if, say, one was to donate a brain, exactly what would it be used for? Would it be sent abroad at all? Do you share the uses of the brain? And what happens to parts of the brain that you finish with? How are they disposed?
So the tissue that comes in is basically, once it's been neuropathologically diagnosed that it was from a Parkinson's case or something, is we run an open access tissue bank. So we do get requests, majority of which are from the UK, but we do have some requests from countries that aren't as fortunate to have funders like Parkinson's UK who fund the tissue bank. So some of the tissue actually does go abroad, but the projects are still assessed by the independent sort of management board. So all the applications for tissue that come in get fed to the board that oversees the tissue bank. They assess whether it's a good project, whether it's going to be beneficial for Parkinson's research. So the idea is that we should be supporting Parkinson's research no matter whether it's in the UK or whether it's abroad. Now, you're probably aware that parts of the brain that is affected in Parkinson's disease are very small, like your substantia nigra is about the size of your thumb. So if you've got many researchers after your thumb, it doesn't go very far. So some bits, the key bits, which are affected by Parkinson's, in our early cases that we collected, are almost exhausted. However, there are some studies that they don't need to look at the substantia nigra. They may want a bit of cerebellum or cortex and things like that. So those cases are still useful, but eventually we get to a point where most of the usable tissue has been used up, but we still can keep what we call a few archival blocks from each case, just in case anybody wants to do genetic studies. There is always some tissue left available from those. And any tissue that is then left over at the very, very end, what we do is we use a special company that has like a funeral service for that tissue. So it's disposed of in a very sensitive way. So we only do that when there is basically nothing else that we can use in the brain. But as I said, we still keep some blocks, forever, just in case anybody wants to do genetics or anything else where it doesn't matter whether it's cortex or whether it's cerebellum.
Just as a person who creates the brain bank, I would like to ask your opinion. There's still considerable debate about the actual progression of Parkinson's and related diseases within brain areas. I mean, even the Braak staging and stuff, and some people even state that it starts in the gut, and convinced that it kind of progresses, and the substantia nigra is really just almost like a tiny, although symptomatically important part of the story. What's your insight? Is it in any way conclusive based on tissue results, or what's your current opinion about this?
The Braak hypothesis certainly raised the spectre that then you may have the spread of these altered proteins from one area to another, which may lead to pathology spreading in the brain. But there are a number of groups that don't agree with the actual pattern of spread that Braak hypothesized. In fact, we've just got some tissue for a comparison study from the Michael J. Fox Fund Brain Bank in Arizona. And I asked them, what are your Braak stagings of your cases? And they said, well, we don't agree with that, so we don't use the Braak staging at all. So they are totally against his staging. But certainly there is good evidence that there is a spreading type of pathology. It might not exactly mirror what Braak set out, but quite a lot of people agree with there is some spreading pathology. Exactly what's causing it, whether these altered proteins come out of effective neurons and then infect new ones, that is still a matter of debate. We've been doing some work with Stan Prusner's group in the States, where he's extracted altered proteins from the brains from the tissue bank here, and put them in animals, and you can see some cross-transfection of these proteins causing altered proteins in the mice. So
in particular, we're talking about misfolded alpha-synuclein, or other proteins as well?
Yeah, so we specifically extracted alpha-synuclein from the cases here, but obviously that's in a mouse. So we'd need to, again, get more people to verify that this is the case, and also try it in primates, because years ago, I think even in David Marsden's time, who was a famous neurologist for Parkinson's, talking about 40 years ago, I think what they did was, they did homogenize up some tissue from a Parkinson's patient to put it into monkeys, but they didn't see any transfection then. So.
Okay, so am I right in concluding that basically Parkinson's is not infectious only because we're unlikely to exchange brain tissue, but otherwise it would be?
Now that is obviously, a very...
slightly joking though, but kind of...
but I think it's not going to be infectious like you would normally think of like a virus or, you know, where if
Yeah,
you
I
cut
see.
yourself, you've got a bacterial infection. It's nothing like that. So I think the word infection or
Misleading.
is you know, a bit misleading.
Thanks.
Mechanical stimuli, so is there a lot going on?
Yes, there's still quite a lot of people having deep brain stimulators implanted. The Charing Cross site of our group, you know, the hospital's in the same trust. They implant stimulators there in Oxford and also in a few other centres as well.
Is that uncertain?
Yeah, it's only suitable for certain people. In certain people, once they've done the analysis on the brain scans and things, you know, they realise that in order to target through into the area, you know, they may have to go through a major blood vessel, which is just like impossible. So structural problems are, you know, and also some people, due to their other sort of like physiological condition, makes them unsuitable for surgery.
Is there quite a lot of animals that do you know percentage of people?
I don't know off the top of my head, but it doesn't work for everybody.
No.
You know, there are quite a lot of positive things. You know, you can see, you know, even look on YouTube and you can see some very nice videos about where deep brain stimulators work for people. And the idea is this drug would work exactly in a similar way to the deep brain stimulators, but then it would be open to everybody. And also, you wouldn't have the hazards of going through surgery to get the same results. Yeah. So, because the deep brain stimulator works on the same principle. Basically, if you overexcite these neurons, they'll shut down, and so they'll release less glutamate. And so the idea of these drugs is that they will dampen down the glutamate release from the same cells. Yeah, you've got to have your battery changed every couple of years. And, yes.
So it would be less tablets, would it, this, if you could do this latest new drug?
Yes, yes, yeah. So, well, we don't...
Hopefully without having to get somewhere, have another half, or take another pill, like, you know, like similar. Sometimes the pill decides not to, but you can take a pill and it just, it's, if you knew you could put a, if you could just, you could be like do a dipstick and say, oh, I need that amount, you know, they never measure. You know, if people that have got it like you now, I don't mean people that at the beginning that just slowly started, I just started to got Parkinson, but people that have obviously it's progressed, you know, if you could just find somewhere, or somewhere where you could measure it, you know, the bit goes like from this, you can't,
it's very difficult.
That would be nice.
Yesterday at the Vauxhall Trade Day, someone produced a paper about an earthquake in Italy where 14 Parkinson's patients have been a nasty surprise like everybody else, but their symptoms disappeared for quite a long time, in some cases 25 months.
25 months?
Has anybody got any explanations? I've heard of this before.
Well, if anybody's been in a building where you've had a true evacuation because of a fire, which I have, the Parkinson's people were not the last people out the door. So, so if you could, you know, there are some methods of trying to train other pathways into the brain. You know, people have, you know, marched to music that's being played to them or a metronome, but unfortunately you need to keep changing the song.